So, what is mitosis and why is it important? So, as a lot of people said in the Google form that we filled out before our lesson, mitosis is the division of cells, right? It's how one cell creates another identical cell. It's important to remember that in mitosis, we're creating identical cells. If the cells aren't identical, something has gone really wrong. During mitosis, like we said, a single cell divides into two identical daughter cells. And the major purpose of mitosis is to replace cells that are worn out and for growth of an organism. So we keep using our skin as an example. So our skin is a great example of cells that are worn out very, very quickly. Our skin cells need to be replaced all the time. We always have skin cells regenerating. So that is an example of a cell undergoing mitosis to replace another cell that's been worn out. And an example of growth of an organism is, a little kid. You don't wake up one morning and your kid is 6 foot, 6'3". It takes time. Those cells have to undergo mitosis, the muscles and the bones. All of those cells in their bodies have to multiply and create identical cells for the organism, i.e., your child, to grow. So mitosis occurs in stages. When we're talking about mitosis, though, it is a very, very small percentage of a cell's life. Something like only 10% of the cell's life is spent in mitosis. Most of the time, our cells are in interphase, which is where they are preparing for mitosis. I say here they're just hanging out, but they're not just hanging out. What they are doing is just doing their normal day-to-day functions. You can see here we got 1, 2, 3, 4, 5, 6, maybe even 7. This one looks like it may be starting mitosis, but 5, 6, 7 out of these cells are in interphase. This cell and this cell, and maybe this cell, are the only ones we can really see that are in mitosis. And that just goes to show that most of the time cells are just in interphase. They only spend a very small fraction of their time actually physically dividing. So let's talk about the nucleus. We know that the nucleus holds DNA. We've talked about that for weeks now. That nucleus holds our DNA. During mitosis, all of the DNA in a cell must be duplicated. And that's because we're creating two identical daughter cells. So we need two copies of everything, right? If we don't have two copies of everything, the cells won't be able to form fully. And some other processes may need to occur where these cells are taken care of. All right, so when we are discussing the cell cycle, so not just mitosis, but an entire cell's life, we refer to the cell cycle. So our cell cycle has 4 stages. We have our G1, our gap 1 or our growth phase. We have our S phase, which is DNA synthesis. And we have our G2 phase, which is the growth and preparation for mitosis further. And then finally, that leads us to mitosis, which is cell division. So you can see here that it's cyclic. So once one ends, the other begins. Cells can pause at any time during this before they get to mitosis. If they're not ready, if a cell's DNA is not duplicating correctly, it will be gotten rid of. If the organelles aren't duplicating correctly, there might be something wrong with it, and in that case, it will be gotten rid of. So some cells never make it to mitosis, But if they do make it to mitosis, make it to cell division, they will have undergone these 3 stages first. Now we call G1, S, and G2 phase our interphase. Together, collectively, those 3 phases are called interphase. So if a cell is not in mitosis, a cell will be in interphase. Okay? So when we're talking about our cell cycle, remember we're talking about all of the actions. So we're talking about interphase and mitosis. So in interphase, first of all, we have our G1 or our gap phase, gap 1. Here our contents are duplicating. The cell is increasing in size. Next, we have our S phase, where DNA is going to replicate, but it's called S phase due to the synthesis. That means it's the making of DNA. We're synthesizing DNA. We have our G2, our gap 2. The cell's continuing to grow. Organelles and proteins are ready. They're developed. They're getting ready to divide. At this point, our cell is ready to go into mitosis. The cell is ready to divide if we've reached this G2, the end of G2. So if we've reached the end of G2, our cell will go into mitosis. So mitosis, as we know, is cell division. The end result of mitosis is the formation of 2 identical daughter cells, okay? So these daughter cells are identical, like I said, to each other. They're clones of each other, which is what we want. And this process is followed by cytokinesis, which just means the cell splitting. So that's our cell splitting. Okay, so now let's talk about mitosis. So we talked about interphase, we talked about G1, S, and G2. Now we're going to go into mitosis. In mitosis, the first stage of mitosis, the first phase, is our prophase. So after the cell is done with interphase, it will enter the first stage of mitosis, which we know is prophase. During prophase, the chromatin, so with an IN, chromatin, that DNA, that is what we're calling the DNA right now, that DNA will condense in the nucleus and it is going to form clear, very visible, not to the naked eye, but under a microscope, it's going to form visible chromosomes. So our chromatin is going to condense into chromosomes. Our nucleolus disappears. So remember, in our nucleolus is that high concentration of ribosomes. So we don't need that at the moment, so it's going to begin to disappear. Our nuclear membrane will begin to break down. So the envelope, our membrane of our nucleus, is going to begin to break down. We don't need a defined nucleus right now. We have loads of DNA, right? Our DNA has doubled. So we're going to need more space. We're going to use the whole cell now for mitosis. So we will be able to see in the later slides that our chromatin is going to coil very, very tightly. And that tight coiling of those chromosomes is what is going to form, that tight coiling of the chromatin is what is going to form those visible chromosomes. And we will talk about this one in the next slide, I believe. So when we're talking about chromosomes, like we start with our chromatin, which is that DNA, that's our free floating scattered DNA, right? During interphase, the DNA is in the form of chromatin, but during prophase, our chromatin condenses and we call it chromosomes. Now, if you look here, this is just a little visual representation of our chromosome. And this chromosome has already been duplicated. Remember, in our S phase, our DNA synthesis phase. Our chromosomes duplicated, our DNA duplicated, it's replicated. We have 2 copies of everything. So this half here, this half of this X is one chromatid. We call that a chromatid. And this other half is another chromatid. We call those sister chromatids. And each of those is identical. So that one and that one are identical. That one and that one are identical. That one and that one and that one and that one are identical to the one that they're connected with. All right, remember in prophase, that was our first phase of mitosis, our first stage, that our nuclear membrane is breaking down. Our nucleolus is breaking down. Our chromatin, our DNA, the fancy word for DNA, chromatin. Our chromatin is where in interphase it was scattered very loosely. In prophase it began to coil very tightly and form visible chromosomes. Those chromosomes have that X shape that we know of, and those chromosomes are split down the middle, right? And this is a sister chromatid, and this is a sister chromatid. Those sister chromatids are identical to each other. They've been duplicated in S phase. So now we have metaphase. So metaphase is the second stage of mitosis. It is where these chromosomes are going to meet in the middle, okay? They're going to meet in the middle of our cell. These chromosomes are going to line up along this imaginary plane. They call it the metaphase plate. I like to call it the equator, but that's not technically correct. But our chromosomes are going to line up at the equator. We have these centromeres. So we have a centromere at the North Pole and the South Pole. We just say that those centromeres are at opposite ends of the cell. And those centromeres are going to make spindle fibers that will attach to our chromosomes. So those spindle fibers are what is going to ensure that our chromosomes are lined up in the middle along the metaphase plate. Okay? So our centrosomes are at opposite ends of our cell, and our chromosomes are at the middle, and we can remember that this is metaphase because our chromosomes are meeting in the middle. So in anaphase, this is the third phase of mitosis. These are when our chromosomes, our sister chromatids, are going to begin to pull apart, okay? So remember, we have that X shape. I'm going to try to draw it. We have, this is our chromosome. This whole thing is our chromosome. It's made up of a sister chromatid on this side and a sister chromatid on this side. In the middle, there's this little guy called a centromere. This centromere is holding the two halves together. So this half and this half are being held together by this centromere, okay? So during anaphase, our sister chromatids are going to get pulled apart to opposite ends of the cell. So remember that during S phase, our synthesis phase in interphase, that DNA was duplicated. So these guys are identical. So one sister chromatid is going to get pulled that way. One sister chromatid is going to get pulled that way. Our spindle fibers are going to do this because they're going to contract. That contracting, they're getting shorter, and those spindle fibers are going to break that centromere. Okay, so the centromere is going to break, and those two sister chromatids are going to be pulled to opposite ends of the cell. Also, during anaphase, our spindle fibers, there are other spindle fibers that are preparing the cell for elongation So in the background, there are other spindle fibers that are preparing the cell. They are making the cell a bit longer because ultimately the cell is going to divide. So during telophase, the cell is elongated in shape and it will begin to divide. Now, the actual dividing of the cell is called cytokinesis. We'll talk about that in the next slide. During telophase, the nuclei will begin to reform. So the nuclear membrane, that envelope, will begin to reform. And those chromosomes, we can see we have a sister chromatid here, 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 and here. They're all separated. They will begin to decondense. So they're going to uncoil. They were coiled very, very tightly during prophase. Now they're going to relax. They're going to kind of kick off their shoes and lay back. They're going to return back to the state that we call chromatin. Okay, and also those spindle fibers, we don't need them anymore, so they're going to begin to break down. Okay, so our spindle fibers are going to break down, and our chromosomes are going to decondense to become chromatin. So cytokinesis, it begins during telophase, during the last phase of mitosis, but it kind of continues on until after telophase. So during telophase, we know that our nuclear membranes are returning, our chromosomes are decondensing and returning back to chromatin. But during cytokinesis is the process of the cells actually splitting. So we can see that there is this thing in the middle. We can see where they're going to split. This is called that cleavage furrow. So you can see they're beginning to cleave off of one another. They're furrowing. There's a dent. They're trying to split apart. They're pulling, right? During cytokinesis, the cytoplasm of these two daughter cells needs to be split equally. So we're going to ensure that the cytoplasm is split equally because a protein ring in between these two cells is going to pinch the cytoplasm along this line, along this cleavage furrow, and it's going to ensure that each of the daughter cells has an equal amount of things because we want two identical daughter cells. So you can see that those cells have ultimately split. We've got our two daughter cells. They're identical. We have chromatin now in our nucleus. We have fully formed nuclei, and we're good to go. The process is complete. All right, so I have a couple videos here that would be really helpful if you watch them. They're really great visual representations of the stages of mitosis, and they're quite funny and entertaining as well. So check those out. So this is important to note, okay? Before we actually start mitosis, when our cells are in interphase, before they enter S phase, we have 23 pairs of chromosomes, okay? We all know that. We've got 23 pairs, so that would be 46 total. So we get one from our mom for each of these. If this is our chromosomes, I'll draw 3. So we'll say our first set here, we have one from our mom here, one from our dad, one from our mom, one from our dad, and so on and so forth. So during interphase, we have 23 pairs of chromosomes. Now during S phase of interphase, we know that our DNA duplicates. And what that means is that each of these individual chromosomes is going to double, okay? So now that looks like a backwards K, but it's not. I am trying to draw our chromosomes, and here I'll just draw them like that. So our chromosomes are duplicating. So now these two are called sister chromatids. So you have one sister chromatid here, one sister chromatid here. And like we said earlier, they're attached by a centromere in the middle. So these sister chromatids are identical to each other. So what this really means is that during interphase, we have 23 pairs of chromosomes. So 23 times 2, as we know, is 46. My hand can't keep up with my brain. So we have 46 chromosomes normally. But whenever our DNA duplicates, so starting in S phase and all the way through mitosis until our cells split, we have 46 times 2 DNA. So we actually have 92 chromosomes during mitosis, because ultimately that 92 is going to divide in half because each of our daughter cells is going to get 46. So then whenever we have our daughter cells at the end of mitosis, they will once again have 23 pairs of chromosomes. So mitosis is very, very important, right? We need to grow. We constantly need cells to be made. We always need cells to be replaced. In our skin, in our body, in our muscle, cells are always being damaged. Cells are dying. Cells always need to be replaced. So mitosis is very important. And when we're growing, when we're young, we talked about how you don't just wake up one morning and you've grown 4 feet overnight. And I don't mean 4 feet. I mean in height. You don't just grow 4 feet. You don't grow 4 feet overnight either. It takes time, right? It takes time to grow, and that's because it takes time for our cells to divide. So if our cell growth is not regulated, uncontrolled cell growth can result in cancer. So during interphase, cells undergo checks and balances. And if DNA is mutated, if something is wrong with the cell, if something hasn't replicated correctly, if organelles aren't duplicated like they should be, the cell is probably not going to go into mitosis. So the cell will be stopped. And what will happen to the cell is that it will be killed by a white blood cell, okay? So some white blood cells, they can kind of regulate cell growth, which is very important because uncontrolled cell growth, unregulated cell growth can result in cancer. So we call cell death is called apoptosis. Apoptosis. That is when a white blood cell comes along and kills a cell. The definition is regulated cell death. So it needs to happen. Might sound a bit harsh, but it needs to happen.